Good morning again. Remember that I've already completed two 15-minute introductions to functional groups and macromolecules. And just as a little bit of a review, let's go over the proteins. Let's go over our easy way to remember these. So proteins can be structural, they can be hormonal, receptors, enzymes. They can be used for defense, storage, as contractile as far as motor, and transport. And again, at this point, it is not really important that you be able to give a large discussion on how they form structure or hormones or receptors or enzymes, because each of those 8 pieces are brought out at a separate part of the course. I just want to review that SHRED CT is the easy way. And for your first test, you will need to be able to list out the 8 categories of proteins. Usually for the AP exam, you would only need to know 2 or 3 of those to be able to give a good discussion on it. And we left off with, moving down, the 3-dimension, or the ways that proteins are folded as a single fold, a double fold. So it's primary, secondary folds, tertiary folds, or quaternary folds. And those folds are a protein, once it's made at the site of the ribosome, goes to the Golgi apparatus, or may go to the Golgi apparatus, where the folding is checked, and also other pieces could be added to that. So that twisting and the foldings make a unique shape, and they are held together by specific special bonds. And those bonds can be broken down if the pH is altered or if the temperature is altered. And one example of pH, for example, in the human body, the pH is average absolute neutral, except for in the digestive tract, where it's a pH of 2. But, for example, if you take an awful lot of something, alcohol or another substance that could alter the pH, then you could have difficulty with all of the proteins of the body. Again, if you have a high fever, and that depends on your age, so younger children have a tendency to be able, their proteins are much more durable due to temperature, and so if they get the fever of 104, the proteins tend to stay together, and after 104, above that, that's when they start to denature or degrade, and adults don't do quite as well at once you hit above 103. So that sequence of the amino acids, remember the anticodons that are at the ribosome making that polypeptide or that chain, determines what structure that specific protein should have. So for example here, they are showing you an antibody. What all of us are would like to make sure that we all have the COVID-19 antibodies. And so this is actually the folding of that. It's very strange. And here you can see the protein from a flu virus, and it's actually very sophisticated. And so viruses are not considered to be living organisms. They do not meet the criteria for a living organism, and I'm going to emphasize that many times throughout the course. So it's strange considering how much damage it is doing and how many deaths it's doing, but there are criteria to be alive, and one is to be able to reproduce on its own, and viruses can't. They need a host to be able to do that. So primary structure, the secondary structure is found in most of our proteins. So if you were asked about, list something important about a protein, I would list how easily it's denatured or broken down. I would list that the secondary structure, the secondary way it's folded, is the most common. Tertiary is actually folded and interacted by its side groups, so those side carbons to that. And the quaternary is, we have to have multiple polypeptide chains. So here we have a primary structure. Here you'll recognize that would have been the codon had been come in, an anticodon would have been made. Each one of these little beads would have been a separate amino acid, but put together in groups of 3, they start making that polypeptide chain. And this would have been at the site of the ribosome. And then they put on the end of this one of those functional groups, and that's what decides what specific type of protein it's going to be, is the end functional group and what any side side chains that they could have. So primary structure doesn't have any side chains. And we can break those categories of what kind of proteins we have by fibrous. And the fibrous proteins only have they're either primary or a secondary structure. An example of fibrous tissue would be in the human body collagen, and collagen is what holds our skin together firmly in place. It's for structure. So as a person ages, right after about the age of 50, there's less collagen made, so the upper lip thins. That causes wrinkling in the upper, in what we call the maxilla region of the face. The same collagen that would hold up mammary glands or the testicles, that all starts to go away. So, but that's an example of a fibrous protein. Then we have a globular protein, and those again have primary, secondary, or tertiary structures. And although it is uncommon, they could have both fibrous proteins and your globs, your globular proteins that you would find in glands, for example. Both would have, could possibly have that quaternary structure, although again, secondary structure is the most common. So we have fibrous and globular types of proteins. That's one way of categorizing them, and again is by their structure. So the sequence of amino acids is just like the letters of an alphabet, and it is that primary structure is what we get inherited. And again, if I go back to that primary structure, in this group I have a functional end group to that, but I don't have any side groups to that. It's just a single fold. In a secondary structure I have, it's called pleated folding. And so if you can think of taking that piece of paper as you would have done as a child and folding it to make that fan, that is exactly what a secondary structure folding looks like. And they are held together by alpha helix. Those are our hydrogen bonds, or we could have pleated. So the alpha helix you're familiar with. Where have we seen the helix before? And that's in DNA, where you're going to have that single hydrogen bond holding your nucleotide bases together. And in the type B type of secondary structure, you are going to have the pleats like the fan. The second picture they show you there is that tertiary structure, and that is really that folded one on top of the other, and then there will be side chains that help fold it additionally. And the quaternary, it really works completely by what kind of side chains they have folding it together. And at this point, I would just focus in on primary and secondary folding. And that primary has just a single strand fold. And the secondary, you have either alpha helix or your B pleated. And I would be able to say that, again, it's hydrogen bonds that are folding that together. And that's one of the reasons why proteins are so easily denatured, is because hydrogen bonds are fairly easy to break. So again, that secondary structure, we have either the coiled, the alpha helix, or the folded like the fan, the B pleated, excuse me. It's kind of one of the nice examples here of the spider silk. And I actually when I lived in Japan, the silk factory was for the country was in the community I lived in Yokohama, and the silk factory was there, and the silk factory museum was there. And you could go in and you could see all of the little spiders, and with mulberry, that's a plant they particularly like, making the very fine silk to make cloth out of. So that tertiary structure is dependent on those side groups, the R groups that would be off the backbone of that. And again, we would have, I can't imagine that it's going to come up, because again, I don't think they're going to be into as intense covalent content-based. So the tertiary structure is by strong covalent bonds called disulfide bonds. It's one of those only places you're going to find the functional S group there. And so that would be one thing to look at. If there's a folded picture and you see an S as a, S is within that for your folds, that would be a tertiary structure. Here again, we have our S's. So here we have a quaternary, and again, you can have those in either your fibrous or globular proteins, but they're 4 identical polypeptides that have been folded one on top of each other. An example of that is red blood cells. So collagen is one of those, hemoglobin again. And so I will talk a little bit about sickle cell later on in the course, but one of those, it's inherited. And so they told you, if we go back right from the very beginning, the important thing about primary is it's determined by your genetic makeup. And so in sickle cell, the red blood cells are not that nice rounded pieces. They are, part of them are in a sickle, what you would cut the wheat with shape, and they can hook onto each other. So that's just one single amino acid. And they are coming up, National Institute of Health is coming up with they have completed a trial, for a treatment of sickle cell by killing off all of the red blood cells through chemotherapy and giving stem cells from a healthy person into the bone marrow and letting that develop. And so far it's been successful. So again, the primary structure has a single fold. It is genetically inherited. The secondary structures are either alpha or beta. The alpha is your helix. Your beta is in pleats. Both held together by hydrogen bonds. Your quaternary will have side groups that are held together, and those side groups will determine what kind of protein you have. So we've talked about what can affect that protein structure. pH, salt concentration, so too much salt in the body, temperature, all of those are what we call, I've said this several times now, denaturing. That means breaking it down, and they do not fix. Chaperonins are protein molecules that assist in that folding, and so we know that one of the problems with Alzheimer's and Parkinson's is that these are building up in the brain, and so far we really have not come up with a good treatment for that. Mad cow disease, where prions were consumed by mad cows, and they went to their brain and really disintegrated the brain, and that could be transferred to a human if they ate the contaminated cow meat into Jacob-Christoph disorder. And I don't think we need to worry about that right now, or acids or bases. I'm looking for my nucleic acids. So here I have nucleic acids right here. So those I spend very little time on because there's only 2 places we really need to worry about those, and that's DNA and RNA. So deoxyribonucleic acid and ribonucleic acid. So here we have the nucleic acids. The nitrogen base is what makes that the nucleic acid. And again, the nitrogen bases you're going to have were cytosine, guanine, adenine, thymine, or cytosine, guanine, adenine, and uracil. And so that is really the only place that you're going to see the nucleic acids come up. So again, with our proteins today, we're focusing on that there are different ways that that's folded once it leaves the ribosome. Primary, which is just a single fold. Secondary, where we have either it's a pleated like a fan, and that's called a beta, or alpha, where it's a helix, and both of those are held together by weak hydrogen bonds. We then have our tertiary, which is held together by our, and if I go back up here, our tertiary is held together by our sulfide bonds. And remember, in our functional groups, I said we don't have to worry about that much. This is really the only place that it will show up. And then our quaternary, it's 4 identical polypeptide bonds and forms collagen, hemoglobin. And our other is, we've talked throughout the entire thing on how things are denatured, and our 2 nucleic acids that are our DNA and our RNA. And that ends our introduction to the macromolecules. Thank you.